Hello. How is everybody tonight? So many cephalopod fans. So I'm Jenny Staletovich. I'm the environmental editor at WLRN. Welcome. I'm sure tonight is going to be really exciting for everybody, especially me. I love cephalopods. Before we get started, I want to bring up Sheila Rankin and Bonnie Berman with WLRN, friends of WLRN.
Hello, everybody. I'm Sheila Rankin. This is Bonnie Berman. We just wanted to take 15 seconds to thank you for being here and joining us for this fun, exciting opportunity to talk about cephalopods. And again, thank you all so much for your support of WLRN. We love you and we appreciate your intellectual curiosity and the opportunity to entertain you every Friday at 2 p.m. with Ira Flatow in this great program. So I'll turn it over to Bonnie.
Just a couple of words just to say that thank you for being here. Thank you for coming to the meet and greet if you did. Thank you for your support because you made this evening possible. So we'd love to hear from you. If you're members, great. If you're not, join us. It feels great. And enjoy the evening.
And thank you again.
And next, I would like to thank our gracious host. We wouldn't be here if it wasn't for the University of Miami Rosenstiel School. I've spent a lot of time here reporting over the years. I feel like I have talked to every researcher here, whether it was about climate change and coral, atmospheric conditions. I started going over the list of people that I've talked to, and I hit like 10, and I was like, okay, stop. But I'd like to bring up the dean of the school, Ronnie Avasar. I was lucky enough to fly in a helicopter with him. He's an atmospheric scientist, and I will say an excellent pilot.
Thank you. Really appreciate it, Jenny. Always a pleasure to see you here and to welcome you. So it is my true pleasure to welcome everybody tonight to this wonderful event. We are delighted to have Ira and WLRN here with us. A couple of months ago, we had Vice President Harris coming to the school. And, we love that because it gives a lot of publicity for the school. But I can tell you that was, kind of a great honor, but it was not as exciting as having Ira here, okay? We love getting this visibility. The school, interestingly enough, is very, very well known from a scientific point of view. Obviously, marine biology and ecology is one of our departments and one of our flagship at the school. But we do many, many other things. We have 5 departments. So the marine biology is one of them. We have a department of oceanography. And some of you have seen with Dr. Brian House, the sustain that is capable of producing a hurricane force 5 winds and simulate hurricanes in a chamber so that we can study them. We have a department of atmospheric science, we have a department of geology, and we have a department of environmental science and policy. So, we really cover all the spectrum of the Earth sciences. And we currently have something on the order of 260 research projects going on. So, you name it that is related to Earth science, and we probably do it at the school. And if not, we'll write with you a proposal and we will do it. And again, a true pleasure to have you all here this evening, and welcome again. And I look forward to the program. Thank you.
And I know I said we have a lot of cephalopod fans. Actually, we probably have a ton of Ira Flatow fans. That's why we're all here. He is one of those really special journalists who can translate science into everyday language, make us understand why it's relevant to our everyday lives. So without any further ado, welcome.
Wow, how do I follow that? Thank you, Dean Avatar, for those kind words. I am not running, and if drafted, I will not serve. And thank you very much, Jenny. And I am so happy to be here because people who know me know I love the oceans. I used to have a 90-gallon marine aquarium. Well, it was actually a reef tank in my living room, and the salt creep got to be too much after a while. And too much hair algae, and it lasted about 5 years, but I enjoyed every minute of it. So I enjoy talking and being among people who understand and work on the oceans. Because having that tank, I understood how fragile the water chemistry had to be, how fragile it was. If even put something, my arm in the tank, if there was something on there. So I applaud all of you working in marine sciences here who will help us preserve what we have, and I'm very excited to learn more about what you do and inform the public about it. But tonight, we're going to narrow it down a little bit to talk about one of my favorite topics, and I hope it will be when you're done, cephalopods. We've been doing Cephalopod Week for about 10 years now. People ask me, what started? Why did you start on cephalopods? Well, we got tired of hearing about Shark Week. And we wanted to have an intelligent sea creature to talk about. I know I'm going to hear from the shark people. I'm going to talk about. You can't compare the sharks to the cephalopods, I'm sorry, in intelligence and cuteness and everything else. So, pro tip for you tonight when you go home, octopuses do not have tentacles. They have arms. So you have a nice bit of trivia to bring with you tonight when you're having a beer or talking about, guess what I learned? Science Friday tonight, and that's what we're here to do. My first guest is Dr. Lynne Fieber, professor of marine biology and ecology here at the university. She has studied the nervous system of all types of marine invertebrates, including sea slugs and our favorite cephalopods. Welcome to Science Friday.
Thank you, Ira.
Also with me is Dr. Andrea Durant, postdoctoral fellow in the Grosell Environmental Physiology and Toxicology lab, who is currently studying how tiny glass squid are faring in a rapidly changing ocean. Welcome to Science Friday.
Thank you for having me.
Lynne, you've worked with cephalopods and other mollusks for many years. How do you conduct research with the animals? I mean, what are you trying to uncover?
Well, Ira, the nervous system of all animals is basically the same, from squid and octopus up to humans, but also down to snails and very other more simple forms of life. And so if you want to understand how the nervous system operates on an elemental level, on a basic level, like the level of cells, or even the level of molecules, working in simple marine organisms is actually a very good way of approaching these ideas. And so if you wanted to know how learning happens, or if you wanted to know what a memory is, like if you could hold it in your hand, what is it? Working in a simple animal where you can actually have just a few cells, a few nerve cells that are called neurons, responsible for a behavior, then that's an advantage. It makes it easier for you to understand what causes behavior, what causes learning. And so what I do is I take the elemental way in which nerve cells communicate, and that's electricity. And I listen to nerve cells talking to one another in the brains of marine animals. And I do this with some sort of sophisticated recording equipment, but basically, in a dark room, I just listen to the brain talking to itself. And it's a tremendous amount of fun.
Wow. And in fact, I remember when Eric Kandel won the Nobel Prize for working with.
Absolutely.
He actually recorded single cells. It's possible to do that?
Oh, yes, absolutely.
You can listen to one cell?
You can listen to one cell.
How do you do that? You have a probe that's tiny enough?
Yes, you have a little glass electrode that's filled with seawater, and you poke it into the cell. And then you have some sophisticated recording equipment, very sensitive amplifiers that can pick up the signal. And if you want, you can look at it visually. You can look at squiggles on your oscilloscope. Or you can translate it into music and listen to it.
No.
So honestly, when a cell is usually when a nerve cell is talking to another nerve cell or to a muscle and asking it to move, it will do so by means of a certain electrical signal that we call an action potential. And you can just like you would use pseudocolor in a map of a hurricane approaching South Florida, you can assign tones to that action potential. And so without you even looking, you can hear the cell respond to a signal that you deliver to it. You can talk to the cell and you can say, I'm going to ask you to tell me how you communicate with other cells in the brain. And then I let the cell respond, and it'll do so with a tone that I have prescribed. And it makes it easy to teach the electrical medium of nervous system communication to students and
Wow.
to anybody who's interested.
The songs of the cells of the cephalopods. I think
You
we've
have a
got
name.
something here. Now, Andrea, your research doesn't focus so much on neurobiology, right, but on the physiology of a small cephalopod called the glass squid. Tell us what is a glass squid and what do you do with it?
Yes, a glass squid, they're actually quite abundant in the pelagic and deep sea. You would hear them as cockatoo squid as well. And they actually look a lot like jellies, so I don't think a lot of people realize they're looking at squid when they see them on images online. There's a lot of really great ocean exploration that shows them in action in the deep sea. These are really, really cool animals. They're really abundant. There's many different species of these crankid squid, or crankidae is what they're called. And what I'm really interested in is they use a really unique strategy for buoyancy, and that's very different than the shallow water squids that you're commonly probably used to seeing. Octopods, or octopuses, use a very different strategy as well. They crawl and swim on the bottom. Shallow water squids are actively swimming and use jet propulsion like many cephalopod species, and it's an elaborate funnel locking mechanism, and it's really, really interesting. But these squids are fascinating in that they do things very differently, and they hold on to a waste product and basically create a fluid that is just a little bit less dense than seawater, and that accounts for their body weight and gives them lift. And so what that's known as is neutral buoyancy, and they
Right.
basically are the same density as seawater, and that really is an advantage in the deep sea because they can use less energy dedicated to swimming and such.
Yeah, if you're a scuba diver, you know all about
Exactly.
neutral. So they're really old-time scuba divers is what
Absolutely,
they're doing.
yeah.
How did you
Yeah.
get interested in this? Were you always working with them?
Yeah, so one of my advisors said I would make everything about ammonia, and this is true, and it seems to have followed me to the Rosenstiel School. The bread and butter of what I do is really looking at how animals use this waste product, how they excrete it, and there's a lot of peculiar ways that animals use ammonia. So ammonia is produced when metabolically active cells are digesting protein. So really, essentially, all animals and all cells in animals are routinely producing ammonia. And this is a common waste product. Humans excrete it as urea, so your urine is very high in levels of urea. Aquatic animals typically can just get rid of ammonia as it is. It's not toxic in an aquatic environment unless it accumulates to high levels in the body. And so most aquatic animals will flush ammonia out of their gills or gill-like structures or in the urine or in the feces. And these squid decide to hold on to it in really high levels that would kill probably most cells and organs and animals in general. And they do this in a specialized chamber that they possess that is not seen in many squid families. And yeah, so I'm really,
Fascinating.
yeah,
I can see why you're interested in that.
yes, yeah.
No, I mean, it's fascinating. You know,
I will say that it's a strategy that about half of squid families use. About 14 families of squid use this, mostly in the pelagic and deep sea environment. And it's broadly accepted as a strategy for buoyancy, commonly found in even giant squid that house ammonia in their vesicles and such, in their arms or tentacles. And yeah, so these are really interesting in that they really use the specialized chamber for buoyancy instead of
That's
air.
cool.
Yeah.
That's very cool. I know, Lynn, I know that cephalopods are a kind of mollusk, but you work on something that's not a mollusk, the sea slug. Is there a connection, a
Oh,
difference?
yes.
Or?
Sea slugs sea hares are mollusks. So remember, so we've talked about the groups of cephalopods. We've got your gastropods, which are the snails, your bivalves, which are clams and mussels, and the cephalopods. So I work on a type of marine snail that's called the California sea hare, and is sometimes called a slug. It is a marine animal, and it is uniquely suited for studies of learning and memory, because even though it only has a few thousand nerve cells in its brain, which is really not very many, you have 86 billion nerve cells in your brains, and cephalopods have 500 million nerve cells in their brain. So when you only have a few thousand, that is really the sort of brain where it's one nerve cell, one action. So if you have an action or a procedure that you've taught this animal, there are only a few nerve cells involved, and so that makes it a lot easier to sort of track down exactly what the change is in those nerve cells when the animal learns, or when it ages, or something like that. And so that's the animal that I work on,
So you can
primarily.
Yeah, you can apply that to the other cephalopods, the kinds that we're talking about.
Absolutely, absolutely. You know, the general premise that we all work on is that we try to understand a biological concept in a simple organism first. We call that a model. And once we've nailed it down, once we know what that memory is or what that learning is, then we test out our hypothesis on a more complex animal. And whereas a sea hare can be had for about $20 after we rear it here at the Rosenstiel School, testing it out in a squid or an octopus would be a fairly intense job, and squids and octopus for research are extremely hard to come by. They are intelligent animals. We don't want to mess with them, number one. They are capable of such complex learning.
Give me an example of that.
Okay, so
Yeah, so we understand how.
yeah, so well, one of the most astounding things, I think, when you think about an octopus's capabilities for learning how to hunt and how to match its background and how to interact with its environment, is that it learns these things, it knows them as soon as it hatches out, right? I mean, they hatch out as little miniatures of the adults. They go through their larval stage in the egg. And they only live for a year, so they have an awful lot to accomplish in that year. They have to hunt and explore their environment and find mates and breed and then die. And so when you are, if you want to, in an experimental sense, in the laboratory, for example, you might teach an octopus that it's advantageous for you to understand your system if you taught the octopus to not attack a new stimulus in its environment. And that can be done. You can teach the octopus to not react. Its normal tendency would be to immediately attack a new stimulus in its environment.
Something in its tank?
That's right. And by attack, I mean go after it,
right.
Handle it, try to see if it's edible, of course. You always want to know that, if it's edible, especially if it's something new.
A lot of us like that.
Right, absolutely. Of course, if it's a threat, you want to be proactive, right? You want to be the master of your domain, and you want to actually address that threat head on. So you can teach the octopus to not do that. And it isn't easy. It isn't easy for the octopus to restrain itself from that normal impulse. And you do this by either giving a reward or a punishment. And then you can test out the concept that you have brought to that animal model from an earlier animal model with just a few thousand nerve cells, and test it out and find out now if you've got a nerve net with thousands and tens of thousands and hundreds of thousands of neurons trying to communicate with one another to enable that octopus to not act on its instinct and attack that new stimulus, then you can learn more about that process. And of course, these things are conserved right on up the family tree in primates like ourselves and all other animals as well. So this is the beauty of animal models from the marine realm particularly, because you've got so much potential there.
Wow, that's an interesting story. I didn't realize how long, it would take to do that. I didn't realize that an octopus lives a year. It has a lot to do in that 1 year.
Yes, and what I like to tell students is that if octopuses lived for a dozen years or 80 years like humans, they'd take over the world. They absolutely. There'd be no stopping them, obviously. If they had that much time to consolidate their knowledge, they'd be unstoppable. If I could, one other thing, which is just fascinating to me, is that the cephalopods and all invertebrates that we've been discussing so far, these are animals that don't have a spinal cord, and so they have a brain that's a little bit different structure from vertebrates like us, and dogs and cats and whales and sharks and other fish. And the idea that cephalopods have such sophisticated capabilities for learning and for exploiting their environment, interacting with it, we have begun to think that when it comes to cephalopods, they actually have almost now that we're a mature Earth population, they have almost what we could consider to be a parallel evolution for the way their brain works and actual consciousness. And we didn't used to think this. We used to think that all invertebrates were just the precursors of the vertebrates. They were just, various models that evolution came up with before the real event, primates and that sort of thing. But now we've come to think of the sophisticated invertebrates like cephalopods as actually having almost a parallel evolution to their way of dealing with the world. And that's an extremely exciting concept, to have two chances, invertebrates and vertebrates, to figure out how to reach an intelligence, in everyday life.
And maybe take over the world if they were. I see this made-for-TV script going on right now, invasion of the octopuses. Okay. Andrea, I know your research subject, the extremely tiny glass squid, and I can't stress this really enough. They're really tiny, right? Does this make it hard to work with them in the lab if they're that small?
Great question. I think that with my background from dissecting mosquitoes and Daphnia, which are tiny little crustaceans in freshwater systems, from my perspective, no, it's actually a breath of fresh air.
They're pretty big.
They're ginormous, and I can dissect them with the naked eye. And it's opened up a lot of tools for me to really explore aspects of ammonia and ammonia-mediated buoyancy in this animal. Along the lines of what Lynne said about tractable models, I think this is a great example of a species that can be used to understand something that's broadly acknowledged, but from a mechanistic viewpoint, we have no idea how exactly these animals produce and retain this much ammonia and how they don't die in the process of retaining this ammonia. And this can be really applied to all the species in the deep sea that utilize this strategy, which is many of them. It seems to be a parallel strategy that arose for buoyancy that really is great for mitigating energy and other things. So,
do you have to be like a surgeon with tiny little tools?
Yes, we have micro tools that we use. The forceps, the scissors that I use are this big. Everything is underneath the microscope, so really teasing apart the different organs and such is a task, but it's an interesting one. But yeah, I feel like a mini surgeon. I usually joke that I could just perform any surgeries after these. So,
well, we sort of have a doctor in the house.
Yeah, exactly. Yeah, if you don't have a spine.
I'm not even going to go there. Let's talk about the ocean itself and how, Andrea, I'll ask you also, Lynne, changes in the ocean chemistry and temperature might affect these animals.
Yes.
And I'm talking global warming, climate change, things like that.
Absolutely. It seems to the contrary, cephalopod abundances, at least squid, are booming right now. This is thought to be due to a variety of reasons, I think, from overfishing of a lot of the predator species that would normally eat these squid, they're really thriving in a warming and warmer ocean. Species that were never found year-round in the Arctic are being sampled there year-round. So we know a lot from historical records how their abundances are changing. And there's now residents in northern climates that were never there.
Do we know why, or suspect why?
Yes, I think that what's known so far is squids in general really phenotypically plastic is how we call it. They can respond biologically very easily to changing ocean conditions, even in an individual's lifetime. And
Soon they'll live 8 to 12 years. Just watch
they just need a day or two to adapt through. So salinity, temperature, hypoxia, so decreasing dissolved oxygen in the water. Ocean acidification does not really seem to affect. Yeah, so there's not a lot of studies, but in terms of metabolism, they do just fine. Some deep water octopods. Yeah, so from that perspective right now, they're actually extremely abundant.
The other cephalopods too, or just the octopus?
My knowledge is restricted to squids, so I can't speak to and I will say that coastal species are going to experience more exacerbated changes, and those are shown to have some effects. So, but pelagic squid right now are doing quite well. I think their biggest concern is, or biggest threat is deep sea exploration. So a lot of mining and for oils, minerals, gas, the risk of spills, which drew me to Martin Grosell's lab with the Deepwater Horizon spill in the Gulf of Mexico, and how it affects, in my case, deep sea animals, such as these squid. That seems to be the most imminent threat, and we don't really know a lot, if anything, about the biology of these squids, let alone how they're going to respond to these changing conditions. So really, that's where I'm starting, is just to figure out how these animals work, and then apply some of these climate change scenarios to.
So we're all just doing a big climate experiment here in the oceans.
Absolutely, yeah.
Lynn, do you have any insight into what the future might be?
I think, just to add to what Andy said, I think the threats to coastal octopuses is from fishing, overfishing. There are many countries have absolutely no regulations about how much they can collect, and octopuses are a very important keystone species in a lot of reef environments, coastal environments. You take out the octopuses and the lobster, two things that people love to eat when they're on vacation, for example, and suddenly you have an explosion of things like urchins that might have in great numbers when they are allowed to really bloom, have negative effects on coastal environments, because they're usually kept in check by animals like that. And so that's, I think, the main threat that we've got in terms of the coastal species of cephalopods is that they're being overfished.
Interesting. Let's see if we can get some audience questions. Here comes the mic. Okay, go ahead, sir. Let's see if that's working.
Hi, good evening. It's interesting you mentioned how intelligent octopuses are, because I know they can solve puzzles and they've even recognized people. And being so intelligent, I normally thought intelligence led to longevity and a long-term life. But why hasn't that translated into a long life for an octopus when they're so smart, but they're limited to only 1 or 2 years of living?
Good question.
It is a good question. So the simple answer is that octopuses have, and I assume the same is true of squid, but I don't know this. I only know it for octopus. They have programmed self-destruction that is hormonal. And when they become reproductively active, which is, let's say, when they're about 10 or 11 months old, their hormones that are not ordinarily coursing through their bloodstream become active, and they literally kill themselves with these hormones. They self-destruct. So the self-destruct is completely a basal characteristic of the animals. And the only way to prevent them from activating this kill switch of their hormonal outflow is to remove part of their brains called the optic glands, where the hormones are produced. And if you do that, the animal will retreat from sexual immaturity and can live another year.
Wow. It sounds almost like what happened to salmon, right? They go through
Yes.
a life cycle.
Programmed cell death that culminates in the death of the organism.
But
It's
they're
just
not-
how they're built.
Wow. So they're not living 8 to 12 years yet. Yes, ma'am.
So I also have an octopus
Wanna get
intelligence
closer, go-
question. I have an octopus intelligence question as well. It's said that self-recognition is a sign of intelligence, and so is it true that octopuses also have a sense of self-recognition capacity?
Absolutely, they do. There are studies, classical studies, that whenever I teach cephalopods, I have my students read about, where you have introduced an octopus to its neighbor in the adjoining cage, glass cages, so they can see one another. They ordinarily, they start to learn their neighbor by saying, "Oh, I must attack you." But then, of course, they learn over time that there's no negative consequences of getting along. And so they become neighbors. Then when you introduce a new octopus of the same species or a female, if both of the other participants are male, you have not only recognition that you of both octopuses that you have introduced a stranger, but they then also they regress from knowing one another, and they become competitors for the right to attack the new individual. And so they absolutely can recognize one another. There's also anecdotal stories that they can recognize their keepers. And actually, this is very true even of our sea hares. When you flip on the lights at 6 a.m., which is when they start their day, their heart rate skyrockets because they know they're about to get messed with by the people who manage the facility. They're going to get a shower and they're going to get, their cage changed and all that kind of stuff. And so there is definitely recognition in invertebrates.
And do they have the same kind of central nervous system that we have, a brain, or is it more diverse?
In octopuses and squids, there is true cephalization of their otherwise very distributed nervous system. Invertebrates have a tendency to have a lot of little brains that we call ganglia distributed all over their body, kind of near where they need to work, because there's no spinal cord, there's no fast sort of relay system for getting communication from the fingertip or the arm tip to the head. But in octopods, there is true cephalization. There's true coalescence of those ganglia into a brain. They even have a cranium, which is composed of not bone but kind of chitin. So they're on a parallel track. They're imitating what we've got as a brain.
Just as a matter of knowledge, you've used the word octopods twice.
Yeah.
Why octopods?
Octopods. So this comes from my invertebrate museum training. When you're looking out at the group of animals that are related, you recognize that there's at the edges of that group of animal there's blurring into other groups, so is it correct to call the animals at the edges octopuses, or are they octopus-like? So that's what the terminology is for there.
Something else to bring home from tonight. Yes, over here.
My question kind of turned into two parts because everybody's had really fantastic questions. So the first question, so it's very small. How you monitor the sea hares, is it invasive or is it like an ECG method?
No, it's a really good question. I'm glad you asked it because we may as well be truthful. I take their brains out. I anesthetize them and I dissect them with teeny tools and take out the brains, and then I break apart the nerve cells so that I get down to the individual neurons. Okay. And then I poke them with my glass electrode.
Okay.
Yeah.
That's just like my own morbid curiosity.
I need to follow up on that question. Because I used to know a very well-known marine biologist and he was a hematologist in North Carolina, and he worked at Duke. And a trawler would go out all the time and scoop up all these fish, bring them back so he could get the blood and study the blood, and he'd have a terrific bouillabaisse dinner that night. Any similarity here to when you...
It's, you know, we used to have a policy in the Aplysia facility where we were growing these animals where new people had to, as an initiation rite, had to taste sea hair sushi. And it's awful. They use pigments from their food and other compounds from their food as a chemical defense. They put it into the flesh, the skin, to deter predators. And that's what you're eating when you sample sea hair sashimi. It's not tasty at all.
know, every year when we do Cephalopod, we always will get a call from a listener about calamari.
Yeah.
We try to get our tiptoe around that issue, but everybody still wants to think of calamari when they talk about it. You had a second part to that question?
So you said when you take away the self-destruction, they only live an additional year. So why is it only an additional year?
So basically the rest of the body, but the brain too, maybe the brain most essentially, since that's what we're kind of concentrating on here, the cells of the body, the organs, they're just not built to last. They're built to last that 1 year. They live fast and die young.
Is that true of squid also?
Yeah, I will say that pelagic squid are some of the most metabolically active animals, and they burn through a lot of energy.
They
and
burn
a lot
fast,
Of, a lot
yeah.
of yeah,
Mm-hmm.
yeah.
But what is the evolutionary advantage to that if you're going to live fast and die young, right? You're not going to be spreading your genes that much.
But they produce a lot of offspring, so.
Especially squid,
Yeah,
my goodness,
absolutely.
yes.
How many offspring does squid?
Oh, hundreds per clutch.
And how many clutches?
I don't know, actually. Yearly?
Half a dozen?
Yeah.
Something like that,
Yeah.
at
And so,
least, minimum.
so how many of those individual offspring actually survive and don't get eaten by?
Great question. Probably very few of them compared to the
So
adults
they
that
help
you would
feed the
see.
rest of the.
Yeah.
Yeah.
The larvae are planktonic, and they basically just drift in the open water column, so great food for a lot of other animals that are in the water column.
The web of life.
Yeah.
Isn't the movie? Anyhow. Yes.
So
ma'am.
I have two.
Please get up to the microphone.
Oh, hello. So my first question is, I know DARPA had been working years ago with squid and octopus on invisibility cloaks. Do you know anything about that?
Well, I don't know a lot about it, but I know that they just developed the first tractable model of actually albino squid that can actually be used to see the internal organs. But that does remind me of Roger Hanlon, who does a lot of the camouflage, which is fascinating. I'll let you probably know a little bit more about his work and the 4 types of patterning that they use.
As you might expect, the U.S. military is extremely interested in this idea.
Yeah.
And they have funded our colleague Roger Hanlon up at Woods Hole Oceanographic Institution to study this. And it's a pretty good model for that. They have chromatophores, as you know. They have reflective layers beneath the chromatophores. They can really do a lot with their skin, not just in terms of color matching their background, but also things like papillation, making little rough patches in their skin to imitate a pebbly bottom or that sort of thing. And if you're starting from scratch, what you're saying, okay, I'm going to need a whole bunch of motors that are going to move this pigment sac in this particular way, or they're going to collapse it so that it disappears. That the cephalopods are really good models for understanding that. I don't know how far they've gotten. I think that rather than invisibility, it's going to be an environment matching kind of strategy there, and I actually don't know where it is at this point in terms of the field.
So this is a stupid question, but
No
to add
stupid
on.
questions.
Oh, so for people like me who will eat anything, any meat, but I won't eat cephalopods. Is there a word for us?
I think we should invent one. Do you have a suggestion?
Acephalopodian? I don't know.
Acephalopodian, that's a good one. I like it.
We have on our website a video of Roger Hanlon stumbling on a cephalopod, stumbling on an octopus that he thought was a rock.
Yep.
If you want to go to the Science Friday website, you can look for it, search for it. It's called "Where's the Octopus?" because it is so well hidden in many places in this film, and he literally gasps out of his mask when he sees this because he had no. And this is an expert on this. Even surprises him, and we can watch this octopus walk across the bottom of the surface, and as it meets other things, change exactly into this other. It looks like a coral in one piece. It looks like a rock. It looks like a piece of wood. And not only can it change its colors, but its texture,
Yes.
right?
Yeah.
Can they all do that? All
Yes.
the cephalopods?
A lot of his work is in cuttlefish,
Yes.
and he's gotten down to a science of, is it 4 distinct patternings? I can't
Yeah.
remember.
At least 3.
Yeah,
that
at least
I'm
a
aware
color
of.
blocking, a speckle.
Yeah.
And
That-
but they can use combinations of it to match the substrate that they're.
And supposedly from the research we've talked about over the years, their skin is so good. It's like a high resolution TV screen. That's how many colors they can make,
Right.
and instantly change just like the TV screen.
Yep.
Go watch this video. Huh? Where's the octopus? Yes, ma'am.
So octopi have captured the literary world recently with books, nonfiction like Soul of an Octopus, and fiction like Remarkably Bright Creatures and Sea Change. So, have you read any of these books? Do you think they communicate well about octopi, or are they hooey?
I haven't read those books.
I have read a few. I'm not sure. I tend not to remember names, unfortunately, of books. But I think that most science writers or popular science writers do a very good job when it comes to cephalopods because their reputations are at stake. And I think there's a tremendous amount of good scientific information about them. So I think you can probably, unless the book really takes a sort of anthropomorphic kind of view of things. I have a few quibbles with my octopus teacher, the video. That fellow had tremendous footage, and he put a story together from his collection of amazing footage, and that octopus showed a lot of emotion that I don't think was really real, but it is a wonderful. These books are wonderful for capturing people's imagination, and from my perspective as a scientist who tries to communicate science to the public, I think these are wonderful. They really are. They're doing no harm whatsoever. Absolutely. I wanted to say one thing, Ira, and that is that Roger Hanlon is a graduate of the Rosenstiel School. He got his Ph.D. here.
There you go.
1980.
1980. Okay, he's just a youngster. Okay, let's go.
Good evening. Thank you for coming tonight. I've read that octopuses have about 33,000 genes, about 10,000 more than humans. What are they doing with those extra 1/3 genes for their advantage?
This is a tremendous question. We've been studying this in my lab, too. They've got a tremendous number of rough drafts in their genome. Genes that can do a job, but they don't have just, 2 alleles. They have many opportunities for that gene to be expressed with small changes, small differences in the protein sequence that's coded for and that sort of thing. And they may not use all of them. They may be, for an animal that has a 500 million year history on this earth, it's probably not a bad idea to have spares. And that's my perception. I'm not a hardcore molecular biologist, but that's my perception. They've got a lot of stuff to continue to play with over historical time to improve their species.
That makes perfect sense. Thank you.
I'm glad you're not calling it junk DNA, as they used to years
I
ago.
don't think it is. I could be wrong, but I don't think it is.
Yeah.
I don't think there's any such thing as junk DNA. It's probably been there. We'll use it 1 billion years ago, or we'll use it 1 billion years in the future.
There you go.
Thank you. Thank you very much.
Yes, sir.
So I know you just touched on the octopus teacher, and that was actually part of my question, but I was wondering, I watched that movie and it impacted me. I stopped eating octopus after watching that film. But I'm wondering what you think, if that had a positive impact on the public perception of octopus, or maybe what your thoughts were.
100%.
I agree. Yeah, absolutely. I've been to outreach events and such where it seems like that's the general consensus in terms of people's approach to that. And even it's kind of revived the discussion on cephalopod welfare in laboratories and how they're maintained and some of the regulations in terms of how they're cared for in a laboratory setting. So.
All right, thank you. And is it octopi or octopus, plural?
Either.
Either.
Either, yeah.
We have octopods tonight.
Yeah. And octopods too, yeah.
Octopus-like.
All right, thank you.
Octopod. Because self-awareness and consciousness, that's always part of the talk about animals and whether they have that, and that makes them closer to human, right? I mean...
Absolutely.
Yep.
So, Andy, you've got something to say about animal care protocols.
Yeah, absolutely. Well, my limited experience about it is in Canada, if I was doing this research, there would be quite vertebrate level regulations in terms of how they're cared for and some of the requirements in terms of enrichment and keeping them. And I think that applies in the U.S. as well, at least for enrichment.
It's about to, yeah. It's starting to take
Well, tell us
a
about
foothold.
what are some of those?
Yeah, so, well, at least in terms of maintenance, they have to have stimuli that are, representative of their natural environment. And I learned you don't have to have that for fish. We do, or other animals. We do in the lab, provide them with whatever substrate
So not just an aquarium of a certain minimum size, which is a generous minimum size, so that they can explore, but things to explore in their
You
environment.
put fish in with them, is that what you're saying?
Well, then to be prey.
Yeah, sorry.
Well, when you have them alongside the tank, they can see other fish.
I will clarify that if you have fish, you don't have to have enrichment, and if you do have cephalopods, you do have to have enrichment.
Right. So,
how you use them, I think in the U.S., you're not required to humanely euthanize them.
It's changing, but currently.
Up until,
yeah.
Whereas in most other, in Europe and in Canada, you're subject to proper anesthetics and euthanization.
Is that because it's a realization they have feelings?
I think just how complex their organs are. They definitely sense pain and.
Pain is one of those things where it's a state of mind, literally. Pain is a state of mind. You don't define an animal as able to feel pain, even though it may, react to a noxious stimulus like being pinched or something like that. It isn't pain unless there are brain regions that can actually process the noxious stimulus, and a lot of invertebrates don't have that.
Oh, interesting. Yes.
I saw this video a while ago. It was like some study or test. It was on some sea creature. I forget what it was, but there's these two doors. One opened immediately and one opened 15 minutes later. And the study was to see if it could have like judgment and understand time. And they put a normal piece of food behind the one that opened immediately and a more desirable one behind the one that opened later. And since you're talking about an octopus being able to recognize people and itself and other octopi, my question is, how complex is an octopus's sense of judgment?
It's a wonderful question, and it is. What you've described could be an octopus experiment. The octopus would excel at that particular task. It's called operant conditioning, delayed gratification. So you pass up an immediate reward for the promise of a better reward later, and even the sea hares can learn that. It's a really essential capability in the brains of animals.
That's the marshmallow test in people, isn't it? Yeah, you know the marshmallow test?
I don't.
Tell them what no, I'll tell them. It's when you give kids a marshmallow and you give them an option. You can have the marshmallow now, see if I get this correct, or you can wait and get something better later.
Like a s'more?
Okay. Okay. 5 of them. Oh, yeah,
Okay.
5 of them. And whether they will actually wait or not, and they make a judgment about that. So the octopuses could do that test.
Yeah,
They're
better
doing that.
than human children, who probably would take the one marshmallow, right? Yeah.
Some of them. Some of them. Speaking of human children. Yes, ma'am.
So I have a question. Like, how do octopuses change color?
Yeah, how do they do that?
This one. Okay. So they have skin that's very reactive and that contains little color sacs distributed throughout the skin, and lots of little muscles around every color sac that either can contract it or expand it. And when it's expanded, that color sac is seen on the skin. And so that's one of the ways that they change color. And they can produce very complex color patterns, like the floor. The floor here isn't blue or gray, it's a lot of different colors, right? And so it's plaid almost. Yeah. The octopus or the squid could reproduce this plaid with their skin by use of this color sac layer that's controlled by muscles, and then some reflective layers underneath that, so that they can shimmer and
They're amazing.
be a perfect thing to dress up for Halloween as.
Dress up as an octopus for Halloween. And you got all those different changes. But so that means that they have nerves that control each one
Yes.
of those colors?
Yeah,
A
so
nerve
this-
for each one.
Yeah, this is one of the reasons they've got 500 million nerve cells, is that's pretty inefficient, actually,
Yeah.
to do it that way. Fish do it with hormones. They say, heck with the muscle thing. Let's just have the color sacs expand or contract by hormonal means.
But that means they can't do it as quickly, I would
Oh,
imagine.
they can.
They can.
Yeah, maybe not. I mean, it's true. Cephalopods are really fast. But there's a cost. There's a cost. They have to have a lot of their brain devoted to a pretty minor thing. Which
Well,
is,
not if you're an octopus, it might not be judgment like that. Okay. I'm sorry. You're next.
Okay, hard to follow that one up. So I have a Florida climate change question for you regarding cephalopods. You mentioned earlier that like pelagic species might not have as many negative experiences with climate change as we might have thought, but maybe coastal species might. What about red tide, blue-green, like big algal blooms, and the seaweed blob that we've heard about all summer?
Seaweed blob.
Yeah.
Yeah, absolutely. Thank you. That's a great question, and I think a lot of that work is still in its infancy in terms of hypoxia tolerance, so low dissolved oxygen that a lot of the sargassum, these seaweed blooms bring, creates really hypoxic environments, and these squid don't seem to do well, high mortality rates. Things like red tide, I don't actually know if that even really is known in terms of the effects on squid, but there's a lot of different things. So warming with shallow water squids, they're continually swimming, so they're actively swimming, using jet propulsion, a lot of energy that goes into that. And so in coastal regions, higher temperatures actually is speeding up their metabolic rates and how much energy they're utilizing. And so it's really actually quite species specific, where cuttlefish seem to be a lot more tolerant to warming and hypoxic events over the loligo squid that you would normally encounter. So it depends, and I think it's still getting underway in terms of what we know about red tides and such, for example.
Thank you. Did you see the video about the octopus that was having a nightmare?
Yeah.
Did you all see this? This was.
Yeah.
What's your opinion? Did you think it was? You don't think it was having a nightmare. Lynn, tell us why.
Well, where do I start? No, sleep is complicated, and invertebrate sleep is less complicated than vertebrate sleep, but it probably cannot extend to, if my understanding of dreams is correct, or nightmares, an octopus would have to be consolidating a memory of an experience that it had, like a bad encounter with another organism at night when it's resting. And I'm just not convinced that I think that it has that capability. I don't think it does. It doesn't have, although it has a sophisticated brain, it doesn't have an amygdala. It doesn't have a hippocampus like our brains. And these structures are really quite sophisticated and allow consolidation of memory during sleep, dreaming, and that sort of thing. And I've seen no evidence that cephalopods have that kind of construction to their brain.
Do you have any idea of what it was doing? If not,
I was going to ask
Andrea?
you the same thing.
Yeah.
I do not.
Yeah.
No?
Either it actually experienced a stimulus at night that made it go wacky, or it had, an abnormality. It had an infection or something. That's my judgment. I could be wrong. It's just a hypothesis.
I like that. I haven't heard that explanation before. Yes, sir.
Well, first of all, Ira, are those socks part of the merch?
Oh, yeah, no. You like my cephalopod socks? I got 2 different kinds here. No, I got these at a place called the Sock Exchange.
Sock exchange. Are they online or?
Are they what?
Are they online? Can I get?
I don't know. I bought these years ago when we started doing Cephalopod Week. So I have I think of three other kinds. I only have 2 feet, so I couldn't wear all my other socks on it. But who knows? Next year. Did you have Thank you for that, noticing my sock.
I have 2 questions. First one is I read a book by Ed Young, An Immense World, where he talks about senses and how humans sense the world and how animals sense the world. Do cephalopods can you tell us about senses maybe we don't know about in cephalopods?
I don't know how strong the evidence is in cephalopods, but in other invertebrates like the mantis shrimp, which is a crustacean. This is the, what is the other common name for mantis shrimp? I can't think of it, but.
Firecracker.
Yeah. They have the ability to see into parts of the visual spectrum that we can't, and they can also see polarized light. Now that I've just said that, I have seen studies that suggest that cephalopods can see polarization of light. This is extremely useful in the marine realm, so you can tell what time of day it is, what that sun angle coming through various ocean layers. So those are visual acuities that we lack. Our eyesight, as far as the animal kingdom goes, is pretty poor. Let's see, they have tremendous mechanoreceptive capabilities. They have a very, very sensitive sense of touch. So rather than having new senses, they've got, sort of supercharged senses that are the same ones that we've got.
Wow.
I'll add to that. It seems like they also heavily rely on taste over things like olfaction that other crustaceans, for example, use heavily. So chemoreception is not as big of an influence versus them tasting as they go along.
Interesting. Yes, next.
First, I want to thank you guys for what you do. I'm a first grade teacher, and I can't tell you how many times I've heard Science Friday on the way home, and then I come back on Monday and I tell the kids all about it. So thank you for all that you do.
Thank you for that.
It inspires the teachers to help us inspire the kids. So.
We are nothing without guests like we have up here today. So we rely on the kindness of smart people.
So my
Because
question.
I'm the dumbest person in the room all the time.
No dumb questions. My question is, I've heard that octopus have blue blood. Is that true? And what purpose does that serve?
Yeah.
Do you want to handle that one, Andy?
Tell us about it.
Yeah, I know they have blue blood, and it's.
Do the other cephalopods have blue blood too? Or just
I'm pretty sure it's.
Oh, well,
yep.
Okay, just getting that.
Related to the heme groups that they have and allow you to.
Instead of
expand on-
iron-based hemoglobin, they have copper-based.
Does that serve them in any purpose when they're living?
Does it
that
serve
help
any
them?
purpose? Well, it's just the way they were designed, so an alternate way of doing things. It means that you can poison land snails in your garden with any copper-containing material, right? So when you're keeping mollusks in culture for scientific experiments or as pets or whatever, you have to be extremely careful that none of your hardware is copper containing.
Yeah.
It's very, very important because it's highly toxic to them.
Wow.
Yeah.
The kids are
Three
going to love it.
things
Thank
we've
you.
learned.
Yeah, hi. Thanks again for this wonderful program. There was a giant Pacific octopus that was kept in an aquarium in Oregon that became socialized. And it was an open top tank, and the octopus would get out at night and play hide and seek with the night watchman. And the guy was about 65 years old, and he was getting a hernia problem from hauling this substantial octopus back to the tank so it wouldn't dry out. Have you heard of anything like that before?
I haven't. I know that every time. I have heard this story, actually, though. I think I saw it on social media. And octopus are really interesting in that, they're becoming trapped in tide pools, trying to capture prey, and they can be air exposed, which is, it's fascinating, actually, that they haven't made that invasion to freshwater and other environments,
land.
Exactly, yeah, because they're clearly capable of, but in terms of socialization, I'm not sure. But,
There was an old story I've heard.
A 1-inch gap under a door and hide in a broom closet.
Yeah, well, what are they, muscular hydrostats, they call them, and they can just mold in any way possible to slide through something like that.
Related to that, I heard stories years ago about a guy who had an aquarium, an octopus in one tank, and every morning he would come in and was a fish missing in the other tank across the room. You know this story? And he stood there one night and he saw the octopus crawl out of the tank, go fishing, and come back to his tank. And stories of octopuses. I heard in Greece that they'd be Greek fishermen. They'd put an octopus in their basket, which is a woven basket, and it would wind up in the lap of the bus driver. So that's how. Yeah, those are true. I imagine. Yeah. Oh, good. I'm glad I'm not telling lies here. Okay.
Thank y'all so much for being here today. It's been a wonderful night. But Lynn, previously you were speaking about operant conditioning and what you do in your lab about teaching these animals tasks or routines or patterns, but then you also say that they don't consolidate memories at night. I know it's getting into the weeds.
Sorry, they do consolidate memories, but I don't think that they could. I don't think they dream. I don't think that the consolidation of memories plays out in that way because they don't have the brain structures that would be necessary for that. This is my view. I could be wrong, and we probably should explore this because if they could dream, it would mean that dreaming, like many other things, can be done in more than one way. And that would be interesting to know, right? That'd be good basic science. So.
I have time for just one more question. Sorry. Yes, ma'am. Step up to the mic, please.
Well, just that story of the 60-pound giant Pacific is the basis of the book, Remarkably Bright Creatures.
Okay.
So, and also in that book, they wrote part of the story first person from the octopus, and the octopus solved a mystery of unrelated people because he could understand that they had the same walking gait. So I don't know, is that anthropomorphizing a bit too much?
Lots of head nodding. Lots of head nodding means yes.
Fun book.
On that. Thank you. You were a great audience in asking those questions. Lynn, I understand that you have brought a few surprises.
I have.
For our guest tonight.
As a reward for sticking around. National Institutes of Health funds a research program here at the Rosenstiel School where we grow sea hares for scientific research. We grow them in families and we ship them out once a week to investigators all over the country so that they can do their experiments on the nervous system mostly. My colleagues and several of my junior colleagues are in the back of the auditorium, and they have brought sea hares for the audience to enjoy. These are not swag.
And not sushi.
And not, yeah, they have to, we've counted them and we have to end up with the same number of animals that we started with. So if you would like to experience a sea hare in your hands, just flag one of my colleagues here that's coming down the aisles. We've got about 25 or so of these, so share with your neighbors and enjoy. If you've got long nails or anything else sharp on your hands, please be careful, because otherwise we'll have a seawater accident.
So we have all these plastic bags with sea slugs in them?
Yes, they all have sea hair.
Don't puncture the bag, is what you're saying. Don't
That's
take it out
basically
and put it in your hands.
That's basically it. So you can see how these animals live in the water.
Why are they called sea hares and not sea slugs?
That's a really good point. Somebody thought they have little sensory structures on the top of their head that somebody thought looked like rabbit ears.
Which reminds me, for a long time, cephalopods thought it had to do with, the name had to do with cephalization, but it's really just head-foot.
Head-foot,
The
yes,
tentacles
head-foot.
on the head.
That's right,
And
tentacles.
the tentacles are their feet.
Well, what does cephalization mean when you say?
Oh, sorry. Yeah, that means consolidation of brain structures into a unitary brain.
Yeah.
Don't forget, we know how many are out there. We have to have the same number coming back. So. Yeah.
You can see the rabbit ears?
Yeah. I guess let's start moving the bags down to the front. Whoever brought them out, please collect them and bring them back and count them. I imagine when your students come to work with you with these creatures, they must be the first time they're getting really close to them, right? And what are they most surprised by when you work with them? I mean, are they like the audience here and they look at the bag full of?
You know, when I first introduce students, undergraduate, college students, or high school students to these animals, they're not seeing them in bags. They're seeing them actually in open-topped aquariums. So I, dig in and pick one up and hand it to the students. And the thing that most surprises them is how slimy they are. I had one little third grader once. She really came up with the perfect description. She said, it feels like a chicken breast that my mom just took out of the refrigerator. Yeah.
That's good.
It's cold and slimy. Yeah.
They don't drop them, do they?
I don't know, did anyone's ink on them, ink in the bags? So that shows their kinship with cephalopods. The fact that these animals produce ink that they can release when they're aggravated, it shows how close their relationship is to octopus and squid that we're familiar with inking.
How far away are they? What would it take
to
put them in with cephalopods?
Oh my, it's night and day, really. I mean, you're talking about 3,000 nerve cells in the sea hares and 500 million in octopus. It's a long evolutionary distance. Despite the fact that, cephalopods reign supreme in the Cambrian, that's how long they've been around.
Wow, that is interesting. Andrea, your research subject might be, I guess, elusive to most people.
Yes.
Can you see them in the wild at all? Or would you know what to look for? Or?
You would have to go on a submersible to adventure in the deep. I hope that wasn't too soon. I guess I will say that this week, I think we've learned that the deep sea has a lot of challenges to living and thriving in it. And these squid handle extreme pressure, extreme temperatures, and they have, over their evolutionary history, adapted to that. And the conditions are relatively constant in these environments, but you have to trawl pretty deep to see them.
Yeah.
The juveniles, they have an ontogenic change in depth as they age, and so the juveniles are actually quite close to the surface, and the mature adults will be deeper dwelling.
How would you go about recruiting new students or people to be interested?
Great question. I do think that it's fascinating that this strategy of maintaining a position in the water column is new and to us in terms of what we conventionally know in terms of using air for buoyancy like fish do. Elasmobranchs use oily livers and such like sharks. And so this is a really interesting and cool. And other animals, I will say, in the deep sea have also evolved this in tandem. So crustaceans, some deep sea shrimp will retain ammonium, but these squid really hang on to 60 to 80% of their body weight in just this fluid. And I think it's just interesting to understand how they came about that and how it works. And I would hope that alone would interest recruiting students. Maybe I'll have to do more convincing. I will say that, I will say along the lines of what Lynne mentioned earlier, even though, and I encourage you to Google what these crinkid or glass squid look like, and they do look pretty pathetic, but they are voracious predators in the deep. They really are a keystone species in the water that they inhabit, and they're predators and prey, so they have an important position in the food web and such.
Why is it so rare that we see the giant squid in, a living one? What makes it so rare?
Great question. I will say that one of the advantages of studying this small squid is because we can't catch these giant squid to understand how they use ammonium for buoyancy, and a lot of what we know is from dead animals that have been captured at the surface. They're pretty deep. The Architeuthis, these giant squid are deep dwelling, and it sounds like they really just surface when they are dead, although there have been recent instances where they've been filmed quite shallow. But yeah, that's one of the challenges of a deep dwelling animal.
And Lynne, what's the biggest challenge in your study? Let me amend that, because I have a blank check in my back pocket here.
How did you know I was going to say money?
I talked to a lot of scientists. Answer's always the same. If I were to give you that blank check, what would you spend it? What do you want to know? What kind of equipment would you like to be invented that doesn't work or doesn't exist yet? What is the biggest mystery for you? And I'll ask that to you, Andrea, after this, that keeps you from knowing more.
I think I would like to design an instrument or a series of instruments that could help me understand, and people are doing this, they're using computational models to try to figure this out, to figure out in a more sophisticated animal, like for example an octopus, how huge numbers of neurons in the brain communicate so effectively, so incredibly rapidly, so specifically. If the range of behaviors that an organism engages in suggest tremendous complexity, and we know that is the case. These behaviors are backed with extremely complex operations in the brain, and it would be very cool to be able to sort of see it all at once. Not just lighting up, but actually know what kinds of electrical, connections these neurons had to one another, so that you could, at a glance, see, all right, there's operant conditioning going on, there's associative learning, there's, this and that, at how those things differ. That's what I would like to know.
Andrea, question for you.
Yes, I would say it would be probably more of a technical challenge in terms of actually capturing these squid. It's very expensive to take these research vessels that are fully equipped with labs and crews, and a lot of the limitations is actually being able to acquire deep sea squid that are below 1,000 meters in depth and more. And so if my name was on a blank check, I would spend it on ways to actually collect and study these squid on board. I've had this conversation where if I could really, the advantages of these squid is because they use a fluid for buoyancy, when you bring them up to the surface, it's not air that's going to expand. So the integrity, and you can actually isn't
That's
changed.
amazing.
Yes, it is. It's a really great strategy, and for that reason, you can actually maintain them alive on board. And really, it's temperature that affects whether they'll survive or not. And as long as you keep them cool, they're happy. And so I envision some sort of device that we can use to study their behavior, but on a ship, and that's as far as
we have a lot of creative people listening, so maybe somebody will come up. And I want to thank both of you for being with us today. Dr. Lynne Fieber, Professor of Marine Biology and Ecology, Dr. Andrea Durant, postdoctoral fellow in the Grosell Environmental Physiology and Toxicology Lab. Thank you both for joining us today.
Thank you. Thank you. Thank you, everyone.
They both joined me here at the University of Miami Rosenstiel School of Marine, Atmospheric, and Earth Science in Miami, Florida. And we have reached the end of our evening program for this evening. Thank you, Miami, and thank you all for coming out. Thank you for welcoming us to your community here in the greater area. And I want to thank again the entire WLRN staff for making us feel so much at home, helping us celebrate Cephalopod Week. And thank you all for being so interested in Science Friday and showing up and being a wonderful audience tonight. Everybody, thank you. Drive safely. We'll see you next time on Science Friday.